Hello and welcome to this episode of Our Longevity Futures. I'm your host, Chris Kerwin, and today I have the pleasure of being joined by Professor Annabelle Singer. Annabelle runs a Singer Lab at Georgia Tech and Emory University in the US, where the central goal of the research program is to understand how neural activity produces memories and spurs the brain's immune system. So thank you so much for joining me, Annabelle.
Thank you for having me.
So today we're mainly going to be talking about a study recently published by the Singer Lab, which looked at the safety of gamma sensory flicker therapy in patients with Alzheimer's disease. So Annabelle, could you briefly put into layman's terms what gamma sensory flicker therapy actually is?
Yes, so it's really quite simple. It's flickering lights and sounds that are at a very precise frequency, so 40 hertz, which is in the gamma range. And we found in our animal studies that entrains neural activity in multiple brain regions, and then that has effects on immune cells and immune signals, as well as reduces pathology in animal models of Alzheimer's. So the stimulation is totally non-invasive, and it's done for an hour a day, every day. In this study, we did it for 8 weeks.
So you claim that you and your collaborators recently discovered that these flickering lights and sounds at the 40 hertz, as you say, it drives gamma frequency neuroactivation in sort of multiple brain areas and reduces this amyloid pathology in mouse models of Alzheimer's disease. Now, where did this, the initial idea actually come from? Because it's quite maybe even an unorthodox form of therapy.
Yes, absolutely. So it started by looking at deficits in neural activity or neural activity that's lacking in the animal models of the disease. And we found deficits in a pattern of activity we call replay, which is like when the brain replays prior experiences to remember them later. And part of that activity is this gamma frequency, for the stimulation, we're doing 40 hertz, but gamma in the brain is a wide range of frequencies, but what we found was missing was around 40 hertz. And so that inspired us to stimulate at gamma and see what the effect would be. And in particular, some of those deficits we found were before we saw memory deficits in the mice. So that made us wonder if this would affect pathology early in the disease, as opposed to affecting memory, which we know gamma plays a role in.
Amazing. And we know sort of what do we know about the biochemical pathways that are stimulated by the 40 hertz frequency, and why do they have such a seemingly positive impact on Alzheimer's progression?
Yeah, so we're currently studying that. What is the biochemical signaling involved? We've found so far changes in immune signals, including phosphorylation of pathways that we know controls immune signaling, that in particular cytokines. So cytokines are signals between cells that we know affect immune cells. And then we know that immune cells change in response to this stimulation. And so the immune cells, in particular, one cell type of interest are microglia. They are like the trash collectors of the brain. They move around and take up things, including pathogens like amyloid beta. So we think they're playing a role in clearing out pathogens. We also see changes in synapses. So those are the connections between neurons. And some of the biochemical pathways we've identified are important for synaptic plasticity, or strengthening and weakening connections between neurons.
So what if this frequency has an effect on Alzheimer's? Is there any other frequencies that have any different effects at all?
Yes. So far in the studies we've published, we really compared 40 hertz to random, meaning there's stimulation, but the frequency is not regular. It's a randomized pattern. And then some other frequencies like 20 hertz and 80 hertz. Most of the time, 40 hertz looks different than all the other frequencies of stimulation. But in some cases, we see differences between the other frequencies. And it depends a little bit on the assay we're using and the way we're stimulating and things like that. So far, I think there are really clear differences between 40 hertz and 20 hertz, and there are clear differences between stimulation and no stimulation, as well as there are differences between random stimulation and the other frequencies. Whether those are all going to be useful in the future is to be determined. I think so. Based on what I've seen so far, and not all of it's published, I think that there's going to be useful stimulation frequencies outside the 40 hertz range.
Oh, that's fascinating to hear. And we recently had news that for the first time in 20 years, the FDA has actually granted approval for a drug that specifically targets Alzheimer's, called aducanumab, and it's a drug that targets amyloid, as you were talking about before, the protein that forms sort of abnormal clumps in the brain, and which can damage cells. Now, is this a completely different mechanism by which flickering lights sort of therapy tackles the disease? And if so, how is it different?
Yes, it's definitely a different mechanism. So as you've probably read in the news, that drug is somewhat controversial. Because the effects on amyloid are clear, but the effects on cognition are quite mixed. And overall, there's a debate in the field whether clearing amyloid alone is going to be enough as a treatment. The reality is Alzheimer's affects the brain at multiple levels. It affects amyloid, meaning there's amyloid accumulation. There's also tau hyperphosphorylation that accumulates into tangles. There's changes in inflammation. There's changes in synapses. Synaptic loss is actually one of the best predictors of cognitive decline. So what I think we need is a drug or treatment that targets multiple levels. It may even be that we need treatments that are personalized, meaning one person might have an inflammatory form of Alzheimer's where really what needs to be treated is inflammation, and somebody else might need a different kind of treatment. So with all that said, we really need to think about the disease at this multi-level way. The stimulation that we're talking about does have effects at multiple levels. It makes it somewhat complicated in that we don't fully understand which level of change or which level of effect is the most important, but it does mean that we're having this complicated multi-stage effect. So we have effects on inflammation and immune signaling. We have effects on synapses and synaptic plasticity. We see effects on amyloid as well as phosphorylated tau. So while we still need to work through whether each one of those things matters for the improvement, for instance, in mice, but also in humans, it's at least promising that we're having effects on multiple levels.
Does this leave the door open for possibly in the future a sort of multi-intervention approach, which may enhance the different, techniques of intervention from their individual sort of impact?
Absolutely. I think first we need a better handle on how varied Alzheimer's can be. Like each person is different. We know that already, but there might be subtypes of Alzheimer's that would benefit from different treatments. And it may be there are some combinations that you would use with one individual versus some combinations you would use with another. There may also be simple drug combinations that are just incredibly effective together. We see this for cancer, where cancer we now know as a much more complicated animal than we thought of it as 40 years ago. And the treatments, we don't have one treatment for cancer. We have many, and each patient gets a tailored approach. I think we're going to see something similar with Alzheimer's in the long run.
And Alzheimer's is fairly famous for having an extremely low translation rate from successful animal testing to successful human trials, and then further to regulatory approval. So are you confident that this sort of unique type of therapy will translate successfully across the human studies? And if so, what gives you that confidence?
Yes, so there absolutely is a low rate of success from animal studies to human studies, not just for Alzheimer's, for many different treatments. We that's part of why we need to do the studies and why we've pushed to get into humans as quickly as possible, because the faster we find out, the better off we are. That said, we won't know, and we won't know. I won't give you a confidence statement until we've seen results from a big, long trial, meaning it doesn't have, hundreds of people over multiple years. That's when we'll know for sure. In the meantime, the results we've seen so far are promising. The results we saw from our own study, I think, are promising, where we see changes in immune signaling and changes in functional connectivity in brain networks. There's some other studies that haven't been published in peer-reviewed journals yet, but they've been presented at conferences that also show some promising results. So I think together, multiple studies showing promising results, it's definitely, I'm intrigued, I'm heartened, but we do need to see the results of a big trial.
And what are some of the main challenges from actually converting it across from an animal study to a human study? Because I imagine with the logistics of actually getting a human sitting down to agree to do this therapy, and the actual, is it a headset or earphones that you have to put on, obviously varies hugely compared to an animal study.
Yes, absolutely. It is a huge challenge. There's so many questions. So first, you asked about the device. So the device is a set of goggles that have lights in them that flicker at the frequency we program it to, and then it's headphones. And people have to do it for an hour a day, which is a big commitment. So it's not a small ask of our participants. But even besides that part, there are so many questions going into the study because mice and humans are so different. So with mice, we did an hour a day for a week, and we thought, well, is that what we should be doing with humans? And we decided an hour a day was a reasonable place to start, but we didn't think a week would be enough. So we opted for 4 to 8 weeks. But there's a whole other question. Should it be an hour a day? Should it be half an hour a day? Should it be 2 hours a day? And that's will be subject of further study. And then there's a whole other question in terms of the assays involved. All the assays we use in mice don't necessarily work in humans. So we have to try to find something that is reasonably equivalent, but will usually in a human participant, we have much less sensitivity in the assays that we have. And then additional challenges, humans are much more variable than mice, right? So we have to try to account for that, for real world variability, both in terms of whether or not they're going to use the device, but also just in terms of their basic biology. So there's many challenges. And in terms of getting this particular study off the ground, it was really a team effort between myself, who had done many animal studies going into it, and our clinical team at Emory, so led by James Law, but also with an enormous clinical staff who makes these studies happen. And we met over and over again to hammer out all the details and go through every every detail of the study, every detail of what we knew from mice, and debated each point that ended up being the study design.
Well, that sounds like the proper way to go about it, sort of debating out each point. And it must have been a sort of a mammoth task to put it on, but it's a really impressive study. And like you mentioned there, having these individuals for an hour a day put on these headsets and undergo the therapy, that is quite a big ask. And how did they actually tolerate the sensory sort of stimulation that they experienced during this therapy?
Yeah, that's a great question. And it was a big concern for us. Would they find the therapy intolerable and just not do it? Or would they, even if it was tolerable, would they keep doing it because an hour a day is a lot to ask? We were pleasantly surprised. We found people tolerated it at a high level, and they can turn the sound and volume and the light intensity up and down. So we do a kind of assessment in the beginning and ask them what they can tolerate in terms of the brightness and the volume. And they tolerated it at very high levels. So that was one thing that was our first surprise. And they tolerated it and they were willing to do it. And then our second surprise, pleasant surprise, was that they adhered to the treatment that they were prescribed. So meaning they're supposed to do it an hour a day, every day, and people did that. On average, they did 95% of their prescribed treatment sessions. I think the minimum was 88%. So they really did adhere to what we asked them to do. We also asked them what the experience was like for them. And anecdotally, I'll paraphrase what some people said. So some of our participants said something along the lines of, well, at first it was hard to find time to do it, but then it became me time. So it somehow, once they got it into their schedule or into their routine, it seemed to take on a positive, or at least not negative, experience of their life.
So I'll go back to something you just said at the start there, was that they could turn down the volume. Do you find that these changes do affect the overall outcome, or is that something you would probably look into? Yeah.
So we did, in combination with the tolerance testing, we do what's called entrainment testing. So we let people tell us what they would tolerate in terms of volume and intensity. And then we made sure that we were entraining gamma neural activity, meaning when they were wearing the device, that produced 40 hertz neural activity. We recorded EEG while they were being stimulated. So we didn't let people turn it down so much that they no longer entrained. And in fact, people naturally selected high levels of intensity, so it wasn't there wasn't really a lot of variability in the intensity that they selected. So I can't really answer that question, and we didn't explicitly test what if they turned down the volume to very low levels. My hypothesis would be if they turned down the volume so low that they weren't in training, that we wouldn't get an effect anymore.
And was this split out? Was this an hour solidly they had to undergo it, or could they split it up throughout the day? How did the structure of it sort of work?
The structure was that they did it an hour a day continuously, so meaning without a break. They could take a break, like say they needed to answer the phone or something like that. They could pause the stimulation and continue, but they were instructed to sit down and do it as a solid hour.
And Alzheimer's is, currently obviously an incurable degenerative condition, meaning that the patients start off with mild symptoms, which with time progressively get worse. Now, what stage of Alzheimer's were the patients at in your study? And what does the data from the animal studies or elsewhere show about the therapy's effectiveness for possibly later stage Alzheimer's?
That's a great question. So our participants were what we call prodromal Alzheimer's. So that means they're early in the disease. They have early memory impairment, but it's not that severe. And they have signs of amyloid pathology, what we call biomarker positive, amyloid and tau pathology. So they're early in the disease. We selected that stage because right now, the prevailing hypothesis is that we need to intervene early in the disease to make headway, basically. So most therapies are really geared towards that early stage. Once you get to the later stages of the disease, it's potentially harder to turn things around. So in the early stages of the disease, there's things like amyloid accumulation, inflammation, and synaptic loss. All of those things are reversible. When you get to the later stages of the disease, that's when you're seeing neuron loss, and that we don't think is reversible. So that's the challenge at the later stages. In between, there's a long period and a lot of different stages. So we've looked at, in our own hands, we've looked at, I would say, early and mid stages of the disease, but it's in a mouse model, the stages it's not really disease stages. It's a little different. And then another person in Liwei Sai's lab, Chinna Adekian, looked at a very aggressive mouse model of neurodegeneration, and he gave flicker stimulation, I think light flicker, for weeks maybe even months, and saw drastic improvement in this aggressive model. He started early, so it's not exactly addressing if you started late, but it was, I thought, quite compelling that it could help even slow down aggressive forms of the disease.
So would you say there's quite a tangible, almost tipping point that once people have put their foot over, then it is hard to rescue? Or is it, like you said, almost a grey area where we're like, well, in somewhere in here, that tipping point happens, but we're not quite sure yet?
I would say it's the latter. We don't know exactly where that tipping point is yet. It could be earlier than, we're currently testing. It could be later than we're currently testing. Right now, the prevailing theory in the field is that it's early. So we need to intervene with our patients as early as possible.
Yeah. And this is sort of a bit more of a broader question, outside the sort of the scope of this single trial. But are there 2 or 3 things that you believe intrinsically are sort of inevitable regarding the future of dementia and Alzheimer's treatment, and for the future of healthcare in general, that you can see coming up on the horizon that maybe isn't tangible just yet? Is there a few things that you think, right, that is going to happen, I'm sure of it?
That's a great question. So I think we're going to, for Alzheimer's, I think we will end up having a landscape, like I said earlier, more like cancer, where we have multiple treatments and we have multiple categorization or kind of personalization of therapies. So I think it's going to be much more diverse. I don't think it's going to be there's one therapy that's the thing for everyone. And I'm looking forward to that. I'm looking forward to there being a slew of options for patients and doctors to try out. And that's one. What I hope is on the horizon are ways to prevent the disease. So not just treating it in the earliest stages, but even before onset. So we would be able to identify people who are at risk of the disease based on a variety of factors, including family history, genetics, health history, etc., and that those people could get an intervention early. That I hope for and I think is possible. As far as the future of healthcare, and this is related to Alzheimer's and neurodegenerative disease in general, I think we're going to see a lot of innovative therapies that are focused on the immune system of the brain. I think there's some in the pipeline. I think there's a lot of science being done in this area. And I think there's going to be quite a bit of innovation in this area.
And for those sort of goals that you see happening, what are the main obstacles that you think that we actually have to get past for that future to be a reality?
Yeah. I mean, there's a ton of basic research that needs to be done. That's number one. And a lot of that's in the works, but still actively, these are very active areas of research. I think number two would be we really need to overhaul the way we do clinical trials. The current clinical trial system is a clinical trial that to prove a therapy is effective is called a pivotal trial, and it's huge and it's incredibly expensive. And so you can't really try many things. You can try, a company, typically with a lot of funds, will try a few things or maybe just one thing. And I think we need more diversity. So to get that, then we need a different kind of trial approach. I don't know exactly what the solution is there, because you do want to be able to test on a lot of people over a long time. But I think that would be really beneficial in terms of getting more shots on goal.
And on this channel, we talk to a lot of people. Obviously, it's focused around aging and health and longevity. And I wonder, obviously, your background in Alzheimer's, obviously, it's an age-related disease, so age has a massive part to do with it. But I wonder, do you and your lab look at different aging research as in the sort of light of aging as a disease? Because obviously we talk a lot about it on here, and I wonder from your background, you obviously don't look specifically at aging as treating aging as a disease, but Alzheimer's, which is a disease of aging. So I wondered what your thoughts were on that sort of almost field of research and how it impacts your work.
Yes, that's a great question. We definitely think about aging for a couple reasons. And we now have studies going or starting looking at healthy aging. So it's definitely an important question. So one of the reasons we are interested in aging is because it has an immune component. So the part of the reason that we think people are more susceptible to disease, but also other things like cognitive loss, memory loss over the course of aging is because we think that there's these immune changes going on as we age. And if we could revitalize our brain's immune system, we think that could be really beneficial. It would both be good for healthy aging, but also be potentially protective against disease. So that's something we're looking at right now. And we're also interested in healthy aging because, like you said, it's a major risk factor for Alzheimer's disease. And if we want to develop preventative treatments, that's where we need to be thinking.
Amazing. And could you give us what the roadmap of where you see gamma sensory flicker therapy actually progressing in the future, and what your plans are for the Singer Lab and future studies that you might sort of, without revealing too much, of course, but whatever you can tell us about the future sort of direction of research, I suppose. Yes,
I think there's a couple major directions. So one is furthering the human trials, and there's a couple of us, as I mentioned, our study's been published. There are some other studies out there that are either in the works or they've presented at conferences. And we plan to get together as a group and talk about everything we've learned to try to really put it all together into a comprehensive picture that'll guide us for the next studies, the next bigger, longer trial. So really the next step there is a larger, longer trial. That is on the sort of human clinical trial front. There's really a lot, though, basic science still to be done in terms of understanding mechanism, like what we know some of it, but there's still quite a bit to understand in terms of mechanism of action, as well as applying this to other different diseases, because there are a long list of diseases that have a neuroimmune component. So if we have a kind of stimulation that controls neuroimmune function, it could potentially be applicable to many diseases. And then another area is developing this, as I said, in terms of a preventative treatment that would protect against disease instead of treating disease after onset. Those are our main areas of interest right now.
Amazing. And where can people keep up to date with your work and the work of the lab?
That's a great question. So we have a lab website, singer.gatech.edu. And I also post our latest and greatest on Twitter. So at Dr. AC Singer. So those are probably the best places to keep up. And then, we present at conferences and try to put our work out there as it comes up.
Amazing. Well, it's absolutely fascinating to listen about and to talk about as well. It's such a almost unorthodox sort of form of therapy. I think it's really interesting to sort of read about and let others know as well. So again, thank you so much for joining us and for giving us your time and explaining everything that's going on at your lab and in your studies. So thank you very much for joining us, Annabelle.
Thank you for having me.
And you can go and check out the Gowing Life website, www.gowinglife.com, where you can check out all our news and articles about health, wellness, and longevity. So thanks again and enjoy your week. Thank you very much.